and introduce Professor Zenon Rajfur, who is an associate professor at the Marian Smoluchowski Institute of Physics at our university, Jagiellonian University in Krakow in Poland. And it's worth noting that Professor Rajfur has held academic positions not only in Poland, but also in the United States, including Rutgers University and the University of North Carolina at Chapel Hill. So it's very impressive with this long-standing academic background in both in Poland and the US. His research received a lot of awards, and his research concerned cell migration, mechanotransduction, and mechanical properties of the cellular environment. The research of Professor Zenon Rajfur lies at the intersection of biophysics, optical microscopy, biosensors, and biomaterials. And he brings this huge, extensive expertise in advanced imaging techniques and biophysical methods, and with a particular focus on developing and applying optical approaches to explore biological system. And we will be talking about optical approaches today. And it's worth noting that his interdisciplinary work connects both physics and the life sciences, and it really supports innovation in biomedical research. We are really delighted to have Professor Rajfur today with us. And without further ado, I would like to give the floor to Professor.
Thank you very much for the kind introduction. And now I should share the screen.
Yes, please.
Okay. Go here and here. Okay, and make it bigger. Oops.
We can see it very well. It's for us. It's a full screen.
Oh, now I think it's better. So it is my pleasure to be here and having this talk to all of you. I will share two recent works which we have done in our group. Those are almost like couple months old works, which I would like to share with you and get the input from you, and maybe interest you in this kind of work. The topic of my talk will be the optical microscopy in studies of cell migration. So let's start with the simple question: why optical microscopy? The first thing, which is very important, is this sentence from 17th century that seeing is believing. Our brain is specifically prepared to work with the images. So the most information we can get is through the images. As they say, one image is worth 1,000 words. So when we see something, we know how it looks like. From the next aspect is that we are living in the environment which is filled with light. Usually we don't notice this, but we are surrounded by light always. So it looks like the light is the least harmful agent which can interact on us, and this is very important, a very important aspect of this methods, because we can observe the biological system in almost physiological state without introducing too much disturbing factors. And another aspect which is also good is that this method seems to be relatively easy to use. Probably if you seen a lot of movies in the TV or other network production, usually there are like two signs that someone is the scientist. Usually you see in the background a lot of glass flask filled with some color liquids, and always there is a microscope somewhere near of this person. So this is like kind of the sign that this person is a scientist. So let's go further with this method. So the development of the microscope started pretty early. This is a pretty old method. It doesn't mean that it is outdated, but the development is pretty long. So we can see like first microscopes from 17th century. We can see, let's say, Van Leeuwenhoek microscope, which is here on the left side. And this is a very simple microscope which consisted only from one lens. And we see another microscope, which is from Robert Hooke microscope, which is more advanced because we can have like two sets of lenses. One is in the objective, one is in the ocular. During the next years, the microscopy started to develop, and in 1950s, 60s, we can see, more advanced microscopes like this, which is shown on this panel. This is we see that there is a lot of improvements here. We can see when we compare with these microscopes on the left, we see that they are mounted on the stable base. They have regulating knobs, stage regulating knobs, where we can move the stage on the XY axis. We can move this also on the Z axis, changing the vertical position of the stage versus the objective. We have advanced light sources, and we can observe our specimen using two eyes, both eyes, which is a great improvement in comparison with those early type of the microscope. This is the microscope which I was working with when I started my career in this field. Luckily, after those years and the development, we come up with this kind of the microscope, which are way more than what we see here. This is actually the photo of one of our microscopes, which we have in our group, and this is not a very simple apparatus. There are a lot of elements which make this relatively simple device into a very complex one. We have like the main microscope here, which is very similar to this microscope which we see on the left, similar in principle, but now we can add a lot of other techniques which also based on the optical principles to this microscope. So we have on our right side, we have the module with the optical tweezers. So we can have our objects here, and we can manipulate them with the focus light beam. On the left side, we can see the confocal microscopy module. So it gives us the opportunity not only to manipulate, but we can also take 3D pictures of the objects which we are studying using confocal microscope. And going further, on the backside of the microscope, we have also a fluorescence lifetime imaging module, which gives us the opportunity to do the lifetime measurements, lifetime of the chromophores, which are excited, and then we can measure also, let's say, Fret effect on our samples. So as we see, we move from this very simple microscope to very complex one. Sharing with you some info, I say when I started the career, I was able to disassemble and assemble any microscope, but now I'm not even trying this because this is too complex. So let's move further. What are the principal light pathways in optical microscope? Usually I'm telling students that there are like two important light pathways. The first one is responsible for the formation of our specimen. And this is the light path which I'm showing here. We have the light source, which is usually the halogen lamp of higher power, like 100-150 watts, which emits the light into this pathway. So we have like collector lenses because we always want to collect as much light as we can. Then we have specialized filters, and one of those filters is the heat filter, which eliminates the infrared radiation in order not to heat our specimen. We can have some another filters like diffusers and color filters, which give us a preferred color of our light. Then this light goes to the condenser, which is very important part of the light of the illumination forming pathway, because it removes, let's say, all the aberration which usually occur during this process, the transmission of the light from the source to our specimen. So many times people neglect the role of these pathways because they say, okay, as long as I can see my object, that's fine. But the quality of the illumination is also responsible for the quality of the image. The basic condition of this pathway is to get the homogeneous illumination of the field of view, because if there is any non-homogeneous illumination, those conditions will get reflected on the quality of the image. And the next important pathway is the image forming pathway. So we have next we have the objective, which is very important part of this pathway. I will talk about this a little bit in the next step. Then the light is transferred to the trinocular, where we can, when it is split on different pathways, we can get the light into the the eyepieces and see by our own eyes, or we can direct the light on the detector, which could be like a CCD camera or photomultiplier or normal photographic camera. Another way to deal with our specimen is to use the fluorescence light, and this will be shown here on this path, where we have the episcopic illuminator. Is similar to this diascopic, but in this case we have light which doesn't go through the specimen. It goes through this same pathway, like image forming pathway, and this light excites the chromophores, which are usually in our specimen. Then those chromophores, they emit the light, which is then again collected by the objective and transferred to the eyepiece or to the detector. As I mentioned, the very important part of our setup is the objective. Basically, it decides about the quality of the image. The one of the important parameter is the numerical aperture, which tells us about the ability of light collecting by certain objective. The higher numerical aperture, then the obtuse angle from which the light is collected is bigger. So that means we can collect more light. And this tells us about the brightness of the objective. Usually when we are dealing with the fluorescence specimen, we tend to use the high numerical aperture objectives because we want to collect as much light as possible. So let's move further. But in my thinking, with my background in physics, I would like to think about the microscope, that this is not a separate device. Usually, when we think about the microscope, we are talking about the microscope setup, which has more elements. So usually, I urge my students to think in the categories of the block scheme. So we divide all the components into certain blocks because this helps us to deal with the possible problems when we work with the microscope. We can have some problems and we have to diagnose them. So I divided all these devices into certain blocks. And first block is the light source. It could be a different light source. Usually a microscope, they don't have only one light source. We have these halogen light sources, which are usually for transmitted light imaging. We have laser light sources, which are used for, let's say, confocal microscopy. And it is good to know where those parts are. This is one block. Then, when we move along this pathway, we go to the stage where usually the specimen is located. But in these times, the specimen is not kept only, in the state of fixation or is somehow not live. Usually we want to carry our experiments on live specimens. So we have to keep this specimen alive. I'm talking especially about cells because this is the level which I'm working on. So we have to keep our cells live. And this is why we have to provide them the conditions which are favorable for them, which they keep them alive. So this is another block. Usually it consists of the environmental chamber, which provides the proper temperature, the proper atmosphere, and keeps them sometimes from the influence of the light. We have this kind of the devices, and this is also very complex system. And in the center of this system is the classical microscope. But on the side we have again detectors, the block of the detectors, because this is not only one detector. On the previous slide I showed the camera, but the detectors could be like photomultipliers in the case of the confocal microscope. It could be diodes, cameras, different kind of cameras. We have, let's say, on one of our scopes, two different cameras, which have to be also taken into consideration. And of course, last but not the least, is the observer, which also interacts with all this system. How we can classify, very short classification of the optical microscope. We can divide them parameters. So let's say the first simple division is the simple or compound microscope. Usually now we have only compound microscope. The simple one, those are loops or something like this. We can also divide them in regard of the optics. We have upright or inverted optic microscope. The upright I shown you on the previous slide, but the inverted is shown on this image. We have in previous slide we have all the illumination system under the specimen, but here we have reverse. We have illumination system on the top of the specimen, and the objectives are from the bottom. This is very useful configuration when we want to deal with the live cells, which are usually kept in the specialized dishes, and there is a liquid which is inside of those dishes, so we can observe them through the glass. We can also have a transmission and or epifluorescent microscope. Usually most of the microscopes works in those two modes. We can also have, when we think about the collecting of the image, we can have a wide field or scanning microscopes. And the example of such a microscope is the confocal microscope. And another types are holographic microscope, which gives us kind of real-time 3D images of the specimen, and specialized microscopes, let's say for opaque samples, which are used in the metallurgical industries or in geological investigation. Of course, we can divide them in many more categories, but this is something which is enough for us. What kind of techniques we can use on the microscopes? Let's start with the transmitted light. Usually, the simplest one is the observation in the bright field, where we don't have any devices which increase the image quality. This is exactly what we can see, and then we can introduce the phase contrast, which increases the quality of the image by showing the edges of the investigated samples, differential interference contrast or Nomarsky, which can give us kind of 3D images of the investigated specimen, or dark field, where especially structures are shown in this contrast. When we move to the fluorescence microscopy, we can also have like techniques which are based on a wide field microscope, and one of those is the total internal reflection microscope, which gives us the possibility to observe only very tiny layer of the sample, just right above the cover slip glass, which is usually like in hundreds of nanometers. And the confocal microscopy, which allows us to get the 3D images of the investigated samples. But optical microscopy also involve using the time resolve methods, where we can deal not in the image domain, but in the time domain. And those methods are like a fluorescence lifetime imaging microscopy, which I shown you before, where we can basically measure the lifetime of the excited chromophores from our sample, or fluorescence recovery after photobleaching, where we can measure a dynamic of the chromophores in our specimen. Another methods where we can also introduce into the microscope, we can use the optical tweezers to manipulate our objects, which is a really clean way to deal with this subject, because we are using clean light beam. We don't have to introduce any mechanical manipulators. We can just use the light. And recent techniques, which are like super resolution methods, like STED, PALM, STORM, or SIM, which can give us much better resolution than traditional imaging. So as we see, the microscopy develops very dynamically. We are inventing new methods which can help us to work on our topics and get new exciting results. And one of the process which I am very interested in is the cell migration, which is not static but dynamic process. And cell migration, this quotation comes from one of the leader of the cell migration consortium, Rick Horowitz, who was responsible for this. This was like more than 10 years dedicated grant from the United States government, where certain groups of the scientists were collaborating in the frames of this project, and the government provide the funding. If I remember well, it was like $80 million spent on this project, and I was very lucky to work in frame of this project. And the cell migration, if we think, is one of the fundamental processes where localized transient signaling in the cell, they have to be integrated with global changes in the cellular architecture. And he also said the cell migration accompanies us from conception to death. Basically, every process is almost connected in some way to the cell migration. Let me show short overview where we can meet or see the cell migration. On the bottom, I show the normal physiological processes which are connected or even based on the cell migration events. We have the action of the immune system. There are like beautiful movies which show how the, let's say, leukocytes chase the bacteria inside our body. Tissue repair is also based on the cell migration. Embryo development, where the cells are moving in groups to certain places in the embryo in order to get certain parts of the body developed. And also wound healing, which is based on the cell migration. I will show the examples of this kind of the cell migration on the next slide. But if sometimes goes wrong with the mechanism which regulates cell migration, we are getting the pathological states. Let's say I listed some of them here, like arthritis, atherosclerosis, osteoporosis, or congenital abnormalities. But the worst one, which the most focus is placed, is the cancer, especially the process of metastasis, where the cells get the ability to move from one part of the body to another and then grow. And this process of metastasis is responsible for the most death in the patients. So we see that the knowledge of the cell migration, and especially how to regulate this process, can have a very great benefit for the humanity. Let's move to the next slide. As usually, we would like to categorize everything. So also we categorize the cell migration in regard, let's say, of the factors which influence or direct the cell migration. And here are certain types of the migration. Let's say a chemotaxis, where the migration is caused or influenced by the chemical factors. We have durotaxis, where the cells move on the gradient of the stiffness of the substrate. Usually cells prefer very stiff substrate. Haptotaxis, where they also respond to the gradient of the adhesiveness of the substrate. And electrotaxis, galvanotaxis, which are connected with the electrical field and the flow of the electrical current. Cells also respond to the oxygen concentration, and this is aerotaxis. Even presence of the water can cause the hydrotaxis. Phototaxis. Some cells are really not happy when they are exposed to the light, and they move away from the illuminated area. So we have different ways to categorize this event. Basically, almost any factor which we can think of can influence this process. What is the classical model of cell migration? So in order for cell to move, what we see here on the left panel is the vertical cross section of the cell. Of course, this is just on a scheme. So first, the cell is sensing the environment, and when it decides to move, it polarizes. That means it produces the front and the back, and then it starts to produce the extension in front. It could be in the form of the lamellipod or some philopods which can go forward. Of course, this extension then has to attach to the substrate by the adhesion complexes, and in order to stabilize this extension. After this step, the cell contracts, which causes the breakage of the adhesion points at the back of the cell, and this causes the release of the rear part of the cell, and as a result, the cell moves into this direction where it wants to move. We see these steps here on this movie. We see, a clear front and clear back, and we see that this cell is producing those extensions, and somehow it has some problems with detachment of the tail, but it is moving into the certain direction. So we see all those steps are repeated in continuous way. So we have some examples of the cell migration, and what we can see here is that many of those examples are not exactly conforming with the classical model. On the left panel, we see fish epidermal keratinocytes, which are one of the fastest moving cells which we can get. They are moving pretty fast. They kept all this nice fan-like shape during the migration, and they move in a kind of the gliding motion. You see how nice movement of this cell is here. The movement is so fast that basically when you are patient, you can see this motion with naked eye. You don't need to record. You see that they are moving pretty fast. Here in the central panel, we can see the example of the wound healing. So when we have a monolayer of the cell and we scratch the wound in this monolayer, those cells automatically tend to move inside of the wound and close this area. And on the right panel, we see a little bit different kind of a motility. This is kind of amoeboidal motility, which is used usually by cells in the very tight environment. There is very few attachment events. They are just moving, producing those kind of the blebs where they move, you know, all the material to those areas. So we see a lot of examples. There are also examples of the growing nerve cones, which also serve as a model of the cell migration. So let's go to the next. And the cell motility is the really complex motion. As I mentioned, during this event, we see the remodeling of the cytoskeleton, and we can see this on this image. We have this fast-moving keratinocytes, and here there is a label of a monomer actin, and we can see how dynamic this process is. So we see the rearward transportation of the actin, and the cell is moving by continuously changing the cytoskeleton. On the right side, there is the image of this area close to the lamellipod, because we think that this is some unorganized chaotic events, but this cell at the edge, it produces very well-defined network of these actin fibers. They even calculate that the angle between two fibers is around 70 degrees. But not only cytoskeleton is being remodeled, but we also see the rearrangement of the adhesion of this cell. And what we can see here, those dark points are the adhesion complexes. And what we can see here, this area will be extending. So this cell will be continuously producing new adhesion points, which should attach this cell into the substrate. So let's see how this works. We can see this extending area and continuous production of this adhesion. And you see that only some of them are strong enough to grow and mature into the focal adhesions. And the same happens here from this side. So the cell migration is very complex process. I think is the one of the most complex processes on the cellular level. Of course, tissue and organisms, those are next level. But even on this level, this is very complex and hard to investigate. But what is even more interesting, what we know that usually those observations were carried on the cells which were grown on the glass substrate, glass or plastic, and those are very stiff substrates. So what happens when we change the stiffness or the elasticity of the substrate, let's say several levels down? So what we see, let's say we have those beautiful rat embryo fibroblast cells here, which are spread on the glass surface, which is very, very stiff. It has around gigapascal Young modulus of the surface. But when we settle them on the elastic substrate, which is way softer than the glass, those cells behave completely differently. They don't spread. They just form those kind of round-shaped cells. So it looks like they are responding to this condition, to the mechanical properties of the environment. And this was long time. People didn't want to deal with this. But when you think about our body, we see that our cells are not living on the stiff glass, which is in that area, but the stiffness of different tissue is really diversified. We can see some neural tissue which is in the order of 50, 70 pascals, which is very, very soft. Lung tissue, 200. Breast tissue, around 1,000 pascals. And going further, we see that different tissues have different mechanical properties. And those cells are living in this environment. They are fully surrounded by this environment, up to calcified bone, which is comparable with the glass, which is in the range of gigapascal. So it looks like we have to take into the consideration of the stiffness of the environment. How to do this? It's really hard to deal with the natural matrix because it's very expensive and really hard to get. So, of course, the scientists try to come up with some model systems. And there are different ways to deal with this, but we choose to work with the polyacrylamide-based elastic substrate. Usually, you see those kind of the material in electrophoresis gels. This is similar. What are the positive sides of this material? Is chemically inert. The cells they don't excrete any chemicals into the medium. It is easy to control their stiffness. We can make very soft substrate or very stiff substrate in the range from single pascals into the hundreds of kilopascal, which is really stiff. In this kind of the polymer substrate, there are very small pores, less than 100 nanometers diameter. So the cells, they don't have this desire to invade into the substrate. And very important from the microscopy point of view, they are optically transparent and relatively cheap to produce. So we are producing this kind of the substrate. So you can see here this photos of this substrate, which we use to observe our cells. And the first example, which I'm going to, so we have elastic substrate, and what to do with this? So we know that cells respond to the mechanical properties of the environment. So what kind of the information we can get out of this fact? So this is a pretty recent work which was done by my PhD students, now Dr. Tomasz Kołodziej, where we investigated how the elasticity of the substrate influences the heterogeneity of the single line WC256 Walker carcinoma cells. So what was the aim of this study? So when we look on the image of those cells plated on the glass, here is on the glass, we see that they are changing shapes, which usually is not we see this many times. Yes, that the cells change their shapes during the observation time. But we try to find out whether they are homogeneous or they are heterogeneous. So the heterogeneity we define as the occurrence of the difference in cell functioning inside of single population. So what we can see here, we can study different aspects, but we decide to go with the very simple approach. We deal with the cell shape or cell morphology. Maybe we can see some laws which govern the changes of the shape of the cell. So we use the time-lapse recording on the microscope. We observe the cells for 4 hours with the 90 seconds interval between the pictures, and kept them, of course, in the proper conditions. So what is the result? When we look on our images, we could divide the cell morphology types into 3 categories: mesenchymal, polygonal and bigonal, and amoeboidal. And this is really nice how we can those apparently very disorganized events categorized into 3 classes. And what we were going to do is to do the qualitative analysis of the parameters of those migrating cells. So we observe a lot of images. We classify each cell on each frame, and what we found out. So when we studied the parameters of those cells, like velocity, turning angle, or elongation, we could collect this set of the data, which in a quantitative way could describe the behavior of the cell. And then we studied this same event on the glass and on the elastic substrate. And what we found first, it was a very important finding, that the cells spontaneously move and change their characteristic. They, let's say, what we see here on this upper panel. For some time, they are moving as a mesenchymal cells, but then they take into the polygonal behavior. Those are not only shapes, those are kind of behavior, because the mesenchymal, they are a kind of moving, producing this lamellipod, and the bigonal or polygonal, they almost stay in the same place and they kind of shake from one place to another. Or we can see another behavior, which is shown on the lower panel. So we can see the cell which moves as amoeboidal. Suddenly they also move into the polygonal. And after this careful observation, we found some rules which those cells exhibit. Usually, the mesenchymal cells, they go to the polygonal or bigonal state, and then from that state they can move to the amoeboidal. There were extremely few cells which move directly from mesenchymal to amoeboidal. So this was very important finding, that we have a spontaneous changing in the shape of the cell, and we can classify those transitions. And when we see that the cells can also spend different time in certain state. From the physical point of view, this is kind of measuring the lifetime of the certain states. So this is like lifetime imaging. So we can see that there are very few cells which are stay in 1 hour state for, let's say, in mesenchymal state for 1 hour or 2-3 hours, that the most stays very, very stable in the state of of the mesenchymal. The same with polygonal and amoeboidal. And when we use the elastic substrate with a 40 kilopascal Young modulus, we see that those lifetimes changes. So the substrate influences the cell morphology and the time when they stay in certain conditions. We also measured how many times during the observation cells move from one state to another, and we found that the introduction of the elastic substrate influences the changes of these transitions. The general conclusion is that when we calculate what is the heterogeneity of the population, we see that on the glass most of the cells are in the mesenchymal state. Then the polygonal, amoeboidal, and there is some small fraction which was hard to classify. But when we settle the same cells on the elastic substrate, the image changes drastically. We see much less cells in the mesenchymal state, but now the population of polygonal and bigonal is really high. And we can see the time course of this of the occurrence of the populations on different conditions. So we see again mesenchymal is really strong, then polygonal and bigonal, they are pretty stable in the time of the experiments. But when we put these cells on the on the elastic substrate, we see drastic changes. The polygonal and bigonal takes over, and the mesenchymal stays much low. So this was to show that the substrate changes the heterogeneity of the cell population. Why this is important? Because up to now, people usually, when they got any results, let's say they measure the velocity of the migrating cells, and they usually average all the results. So we can see here on the right panel, those are the average results from all the measurements. But when we divide on the subpopulation, we see that that amoeboidal has a completely different velocity of the migration than the average number, and the same with the elongation. That there are differences between subpopulations. So what is the conclusion? The first time we observed the spontaneous transition between different population in the cell line, and the elastic substrate influenced the distribution of those subpopulation, but the effect of this substrate is not equal on each subpopulation. And what we show here, these results can give us new insight into the dynamics of the cancer cells, because we know that the cancer cells are pretty heterogeneous, and the role of the mechanical properties of the environment on the heterogeneity of the cancer cells. This may have a pretty big influence on the new therapies of the cancer cells. And because we didn't stop here, we are a kind of really curious people, and we like hard problems. So Dr. Kawajay moved one level up. So now he wants to combine two techniques, which are used in the in the measurement. So we have elastic substrate. So previous work was very simple. We just show how the cells behave on the elastic substrate. But now there is another technique which is very insightful. So this is called attraction for the microscopy. How it works? When we settle cell on the elastic substrate, which we can consider as a set of the springs, so the cell interacts with the substrate and exert forces on the substrate. What is the result? The result is that this cell is able to deform the substrate, and the amplitude of the deformation depends on the forces. So stronger cells can deform a substrate more, weaker cells less. So how we deal with this? So the problem was in how to measure those movement of the substrate, the deformation. So the method is very simple. We introduce the fluorescence markers on the substrate, so we can see when we plate the cell how those cells deform the substrate. So we have the cell which is moving on the substrate, and as you can see, is deforming the substrate, and deformation is read by the movement and changing the position of those markers. And there are mathematical methods which can calculate from this deformation, from displacement of those markers. They can calculate the traction forces. That means they can show which area of the cell produces which force. So now we can tell which part of the cell produces more force. So let's see here. This is exactly the same. Excuse me. This is the same cell, and we can see the map of distraction. So what we can see? We can see that this part produces more forces. It's like more is a stronger part when they move through this substrate. So this is one part, but we made our life harder. So we introduced another factor, which was using the micro and nano diamonds in this research. Those are special kind of the nano diamonds or micro diamonds, where they have certain changes in their crystal structure, and those changes are able to produce defluorescence. So when we have this crystal and then we shine light on this crystal, we see the emission of the fluorescence. Okay, this is interesting, but nothing new. But then we introduce another way. This deformation, or this quasi-atom inside the microdiamond is very sensitive to microwave radiation. So when we apply the microwave radiation on different frequencies to this diamond, it changes the fluorescence. How it works? So we can see this here. This is the spectrum of such a event. So we can observe the level of the fluorescence, which is emitted by this micro or nano diamond. And then what we can see when the temperature around this diamond increases, we can see shifting of the of this central maximum deposition of this spectrum along the frequency of this radiation. So the idea is that this can give us the measure of the temperature, local temperature around this diamond. So we connect like two techniques. We can measure traction forces, which cells exert on the substrate, and then we can measure the local temperature around the cell, which is really not done yet. So the first step was to prepare the proper substrate. So this is a normal vertical cross section of the substrate. We see those fluorescent markers which we've seen in previous images, which are the polystyrene beads, which are fluorescing in green. The problem was the introduction of the layer of the microdiamonds, which have to be on the surface. So we developed methods how to settle them here on this surface. So we have a mixture of the markers of the deformation of the substrate, and we have also diamonds which can measure the temperature in those points. And then, of course, that was the first step. Then we put those the substrate on the microscope stage through the Petri dish with the glass bottom surface. The substrate was here, and we see this here. We have this our substrate, which is green markers and red diamonds, and the cell on the top. And now we have to introduce this microwave radiation. So we have to put the antenna, the microwave antenna, as close as possible to the cells. You see, this is like 250 microns. And then we see that was this part, and whole measurement system is pretty complex. Luckily, we are in physics department, so we were able to connect knowledge and experience of the photonic group with our group of microscopists, and we were able to carry this kind of the experiments. We see those are very complex experiments. We have one layer of the problems over another, and when we solve these problems, we demonstrated that we can measure at the same time two parameters. We can measure the cell traction, that means how the cell behaves or what kind of forces exerts on the substrate, and which part of the cell exerts the forces on the substrate. And at the same time, we could see how the temperature changes in that area. You see, we have the initial peak of this spectrum, and then we see in this red, I couldn't make it bigger. So we see how this peak moves, and this is enough to separate those temperatures. The results, the final results are collected here. So on the red diamonds are the diamonds, the temperature measured by the diamonds, and the blue is controlled temperature measured by the thermocouples. And we see that they are pretty much the same. But what also can you see, the error bars here are much smaller than the error bars on the thermocouple. So we were able to measure the temperature differences with the precision of 0.76 degree, less than one degree Celsius. And at the same time, we can measure also the cell traction. And this was really something which was not shown up to this date. Of course, this method will require more development and perfecting the experimental condition, but we show in principle that we can measure a temperature and then any other parameter which can be measured with the optical microscopy. I think this is everything what I wanted to show, and the final conclusion is that despite of the old age, the optical microscopy is still a powerful tool to study living system. And when we combine this classical optical microscopy with another physical methods, we can carry out the multiparametric analysis of the biological system, because only such kind of experiments can give us the proper information about behavior of the living system. So this is what I wanted to share with you. Thank you very much, and I'm ready to carry out with the questions.